thanks to all of you for joining us today. I'm passionate about connectomics, and I love spreading that gospel whenever I can. I'm a retinal neuroscientist, and I'm interested in the circuits of the retina. I'm interested in how retinal neuron types are wired up together to form the first stages in information processing before you hit the bottleneck of the optic nerve. And so connectomics is a perfect approach for combining with intracellular recording and for light microscopic methods and molecular methods and so forth to try to really break the whole network down and provide a deep mechanistic understanding of how this machine really works. Obviously, connectomics is a complicated process. It involves lots of stages, and my presentation is going to touch on these to varying degrees. One needs to acquire the volume. In my case, that was done by others. The volume needs to be aligned and then ideally segmented using automated methods. Cells need to be reconstructed, partially using this automated segmentation, but not necessarily only that way. We need to identify the cell types within the volume, understand the chemical synapses that can be seen at the EM level, map them all and identify pre- and postsynaptic partners, and then arrive at this description of the connectivity, at least of chemical synapses. The connectome, which is kind of the holy grail we're all after at the end. So the retina is shown over here on the right, and I began my work about 10 years ago with a volume that was generated by Kevin Brigman. Kevin had trained with Winfried Denk, was really at the cutting edge of this whole technology. And the volume that we're looking at here is a chunk of one of the synaptic layers in the retina. This is called the inner plexiform layer here. And this is where the signals coming from the photoreceptor layer carried through these bipolar cells that are descending through the layers of the retina, synapse upon postsynaptic partners, including the ganglion cells here. These are the output neurons. Their axons make up the optic nerve. They're communicating with the brain. So it's all this machinery that I'm interested in understanding, all the cell types and their synaptic connectivity. And Kevin's volume is the first really large volume, about 260 by 200 microns across, and the full thickness of this synaptic layer. But of course, the rest of this circuitry in the outer retina was left out. And this was now stained using heavy metal methods so that you could actually see the synapses here. That was the first volume we began to do our analysis with. There's an earlier retinal volume also from the Denk group that's even smaller and lacks synaptic detail. Some of you may know this volume. I'm happy to talk about that one, but you can see that the scale here is vastly greater. You should be able to see here, I'm hoping you'll be able to pick this up, some herky-jerky steps through the volume. You can see the EM is not wonderful, but it's good enough to see cell membranes for the most part. And with a little bit of work, you can actually reconstruct a lot in here. I'm going to show you some examples of that. So here we're looking at a top-down view of the volume, right? This is a 100 micron scale bar here, and we're looking at a map of the cell bodies in the ganglion cell layer. They include also some interneurons, the amacrine cells. You see the blood vessels here. So this is just a mapping at very low res. You can see that we have lots and lots of skeletons here describing the morphology of lots of different cells, as well as nodes, some of which are describing the locations of synapses that we've mapped. And these are the different cell types that we've identified in the volume. You have to be a retinal specialist to know really what that's all about. But there are, of course, many types of these bipolar cells that are these vertical neurons that are carrying signals from the outer retina to the inner retina into this inner plexiform layer, this synaptic zone. And they're characteristically different in where their axon terminals stratify here. And that gives us a really great way to identify which bipolar cells we've got. in the volume. And if you're looking top down now, you can see that we've reconstructed so many of these bipolar cells that you can see the mosaics of the axon terminals of each of these 15 types of bipolar cells pretty well mapped out by now, which means that if you've got any postsynaptic process getting input from pretty much any any bipolar cell, you can identify its type, and now you're beginning to work towards the connectome. The volume is big enough that even some ganglion cells are small enough to fit within the entire volume. So we can get whole cells, and you can see that these too can form mosaics. They don't always, but often they do. And there are, turns out, we found out that in the very little narrow strip of the inner plexiform layer, there can be multiple mosaics interleaved with one another. And each of these cell types has characteristically different synaptic connectivity. In this case, we're looking at pie charts showing the relative weighting of inputs from different bipolar types. And you can see that despite the similar morphology and level of stratification, field size and all of that, things that would make it very difficult to otherwise discriminate these types, that the input alone would be sufficient to help you make these distinctions. Here we're looking at a large reconstruction of glycinergic amacrine cell synaptic inputs to a class of postsynaptic amacrine cell called the starburst amacrine cell. The starburst is famous for its role in motion vision, optokinetic nystagmus, and things like that. So it's early in detection of motion in the visual system, and that detection is modulated by glycinergic input, but nobody really knew what those glycinergic cells were. We now have reconstructed those cells. We know it's basically 3 types. They have distribution across the whole arbor and so forth. So this is the kind of approach that we've been taking with the Brigman volume. These are all skeleton reconstructions you're looking at here. And this is another type of amacrine cell. This is the VGLUT3 amacrine cell. This has been implicated in a number of different functions, but we went looking for the particular amacrine cell type that might be responsible for the velocity tuning of our image stabilization system. I should really say the mouse's image stabilization system. And that analysis using connectomics identified really only one candidate. We went and found the Cree line that lets us control that cell. When we shut it down, this sort of velocity tuning of the optokinetic system was altered just as we would have predicted from the connectomic insight that drove the physiological experiments. Here's an amacrine cell that's known to be coupled to a glial cell of the retina, the Mueller cell, and we were able to work out its stratification, its synaptic connectivity, not only with the input bipolar types, but also with a variety of other amacrine types as well. Here you see the Mueller cell coupling schematically shown. Here's a top-down view, so you can see that these things also form a mosaic, very much like the bipolar cells or that ganglion cell that I showed you. We've been reconstructing vascular relationships in the retina using this large volume. So this is the whole volume here, and we've mapped all of the capillaries at one level of the retina, and what you're looking at are actually skeleton reconstructions of the pericytes that are the perivascular cells here shown in blue, wrapping the endothelial cell and the lumen of the vessel here. And you can work out the connectivity of neurons with these vessels using volume reconstructions. These weren't done with automated segmentation, but with manual reconstructions, so they're very time-consuming. But you can see here the purple endothelial cell, the blue pericyte. The yellow is points where neurons have direct access to the basement lamina of the vessel here over the endothelial cell. The green spots here are where there's contact onto the pericyte. And some of these pericytes have these spine-like protrusions that go out into the parenchyma, and all of these green, sort of highlighted, light green highlighted zones are areas where the neurons are in direct contact with the surface of the pericyte here. So we don't know exactly what this means, but presumably this has something to do with the neurovascular unit and how neurons can control blood flow within the retina, for example. Okay, so that's all from the volume from Kevin Brigman, K0725, we were eager to try to push into better resolution EM, larger volume, and segmentation. And we did this in collaboration with two wonderful folks from University of Washington, Seattle. Wanqing Yu is a postdoc with Rachel Wong there, and they're experts in generating these volumes. And together, they generated a beautiful new volume, which we call the U volume after Wanqing, which is 250 by 230 by 130 microns. So here's the retina again. Here you see the photoreceptors, their axons coming down, synapsing in the outer synaptic layer. Here are the bipolar cells, their axons coming down, synapsing in the synaptic layer we were studying with the Kevin Brigman volume. But now this new volume spans all of this, right? The Brigman volume was only in here. So we have the full thickness. This is about the size of the Brigman volume. The resolution here is 5 by 5 by 50 nanometer, so 50 nanometer serial block phase sections. Okay, so let me talk just a little bit briefly about the approach to segmentation. Many of you probably know all this stuff. This is kind of the WEBKNOSSOS scalable minds model. You start out with a voxel. The initial CNN, the AI, is going to give you supervoxels consisting of a collection of these voxels, which are the elements that can't be broken any further down in the system. There's going to be an initial phase of agglomeration in which Lego blocks like this white one here, are joined with other blocks of various shapes and sizes using an agglomerate skeleton. So these things are now linked as one element, which is a segment, but they could, in theory, later be broken down. Now, when Norman and the folks at Scalable give me the output of this, I've got a jumbled collection of these segments, and I have to fix the merge and split errors, right? If some of these blocks should be broken into their components, I can do that to make sure that we don't cross across membranes, but I can also merge things together like this to get much larger shapes like this dinosaur. But of course, what we're after are neurons. And so here's an example of a volume reconstruction from this new U volume using segmentation based on ground truth that I produced in the volume, handed to Scalable Minds, which they then used to generate a model to do the segmentation. Now, the point you have to understand here is that Scalable Minds will do this stage of agglomeration for you. They actually give you the opportunity to choose how aggressive to be with this when you're starting with a new data set. And so what you're trying to do is find the right balance of merge and split errors given the relative difficulty of fixing these two so that you're as efficient as you can possibly be. So you have the choice. You can decide whether you want to split them all the way down to the the initial supervoxel size or close to having almost everything merged into a single segment. The last thing I want to mention on this slide is that you can generate from a volume reconstruction like this a skeleton that is just the agglomerate skeleton that just says which of these Lego blocks have to be put together to give you this shape, right? This turns out to be very useful because there's a lot of overhead in displaying a mesh like this, much less in a node edge graph like this. So this allows you to be much more efficient at viewing lots and lots of cells in a big volume, while the GPUs are, limited in the way that they are. So these are some of the kinds of segments that pop out of the process using the U volume, the Scalable Mind segmentation with the appropriate level of aggressiveness of agglomeration without any proofreading. So we get segments that are, 10 microns long, skinny little processes. You can already see differences in the shape of these things, and that can really help you when you're trying to fit split errors. You can see the next piece has the same kind of texture to the surface, which is much easier than when you're reconstructing with skeletons. So we can talk a little bit about the relative merits of skeletonization and volume reconstruction. I showed you that if you have a volume, you can generate a skeleton But you can just go into the WEBKNOSSOS interface and start laying nodes down and generate your own skeletons. In any case, these skeletons give you an overall sense of shape. But if you really want to know the shape of the thing, you want to see the volume reconstruction. Stratification in the retina, that's a really important way of us knowing which cells are which in the retina. Both are pretty good for that. If you want to know where the two cells touch each other, it's a lot easier with volumes than it is with skeletons, although you can use the skeletons to direct you to the right place to look to see if there's contact. Synaptic mapping, the same deal. If you're doing automated synapse detection, you want to know which volume your presynaptic element is sitting in. You can't do that with skeletons, although you can use the skeleton to guide you for online assessment of that in the original volume itself. Skeletons are a lot better for annotation. If you're mapping lots and lots of synapses and you want to say something about every one of them, use individual nodes in a skeleton mode. That's not so easy to do in the volume world. And then as far as speed is concerned, skeletons are going to be much, much faster to generate, but it's getting a lot faster now with the volumes. We're just clicking these chunks of neuron together as we go. So it's a lot faster than it used to be. When I'm working, I really want to be organized about which entities I have and where they go. And of course, in WEBKNOSSOS, there's a whole hierarchical directory structure in which you can insert things. So for me, a chunk of my directory structure might look like this. I have retinal ganglion cells. There's 40 different types in the mouse. But here are 2 of the types. These are actually sort of classes because they're made up of 4 subtypes. Here's one subtype. Here's the first cell of that subtype. I have its skeleton, that's in one place. I have synaptic inputs from all these bipolar cells, which have been identified. Then I have a separate compartment, a separate part of the skeleton, which is just the axonal compartment. Amacrine cells, many types of amacrine cells. Here's the starburst, but they're on and off. Here's just the ons. Here's the first one of those, et cetera, right? So you want to be really organized. If you're going after a connectomic reconstruction of the whole thing, you kind of want to save everything. You reconstruct a little bit, you're not quite sure what it is, you're going to name it as best you can, stick it somewhere in a directory structure to be classified. You're building towards that. If you're doing more directed connectomics, you don't have to worry about that. Just throw it away. Just some examples of the kinds of things that we can analyze this way. These are volume reconstructions of an axon, bipolar axon making en passant synapses. Here's an amacrine cell that's key for signaling the rod signals to the brain. We can reconstruct these and find the place that's probably where it's got most of its sodium channels concentrated. That's another story. We can show you the distribution and the morphology of bipolar cells that selectively contact the cones that are carrying UV, that's like short wavelength visual information. These are some horizontal cell axons. Each one of those little tips is going up into the terminal of a rod bipolar cell. So just to wrap it up here, you have to be thinking about things like, are you going after a targeted question, or are you trying to do the whole thing, right? I'm doing a little bit of both. How do you find your particular cell type of interest? If you're interested in a rare cell type, how do you find a needle in the haystack? Well, in the retina, stratification really helps. It may not in your context. The shape of the cell, how thick are the processes? How frequently do they branch? Do they have spines? Are they varicose? What about intracellular appearance, synaptic connections? And there are molecular methods that would be like a GFP for EM that you could use if you have a Cree line, for example, that would label your particular cell. And then the last thing I want to say is that you can handle cells that are outside of your volume. You can see processes coming in from outside of your volume, and you can see cells with processes that go outside of the volume. And if you're clever about putting those two kinds of information together, you can actually imagine what the cell would have looked like if you weren't just looking through a landscape through a soda straw, but you were seeing the whole thing. I'm happy to talk about that, but I think I should probably wrap it up for now. We have plans for the future. We're going to get automatic synapse detection going in this volume. We're trying to build towards a real first draft connectome with the mouse retina and then refine it through the community. We want ultimately to extend this to other species, look in development, look in disease. And we talked to Norma just a minute ago about the possibility of using AI to accelerate this proofreading process for the segmentation. So I'll stop there and be happy to take any questions. Thanks.